So student, we already discussed about the enzyme action, how the substrate, within the enzyme there is active site, there is substrate binds and ultimately the enzyme reaction occur, and we know the how the at first enzyme and some ES complex formed, then ES complex converted into EP complex divided into E plus P. So we already discussed about that. Now the another part is how there is a graph of potential energy and progress of reaction product. Here in this graph you show that the activation energy enzyme there lowers the acceleration energy of substrate, and in this graph you show that the activation energy is more without activation energy. The graph shows the activation without activation energy without enzyme. The graph shows here, and with activation energy with enzyme the graph is here. So there is the main function of enzyme which lowers the activation energy by particular substrate. Activation energy meaning the particular energy which is needed to begin a reaction, making the substrate active, that is called the activation energy. Now, what are the factors which affect enzyme activity? There are several factors which generally decrease or increase the enzyme activity. As like there is the temperature, pH, etc. Several factors are there which generally maintain the affect the activity of enzyme. Here is the temperature. There enzyme acts at a particular range of temperature. There is a temperature in 20 to 30 degree range. There enzyme works well. But when the temperature increase more, the action of enzyme becomes slow. And ultimately there is a optimum temperature, which is the optimum temperature, the maximum temperature at which the enzyme activity occurs. If we draw a graph here, and we can show the dash like the optimum. This is the optimum temperature. At first the activity of enzyme increase, and that in such temperature the enzyme will reach the maximum value. This is called the optimum temperature. This is happened. Same is case of the pH. In a certain pH limit, the enzyme affect more, but after that limit, the action of the enzyme becomes low. That is called the optimum pH. And why the enzyme activity becomes lower in the high temperature, or enzyme become damaged in high temperature? Because the structure of enzyme is denaturated after a certain temperature. It is made up with protein. We all know that protein become broken down by getting the high temperature. So in high temperature the enzyme become damaged, but in low temperature also the enzyme activity become lower, or the enzyme become inactive. So there is a need a particular enzyme activity, particular temperature or particular pH in which the enzyme activity is more. Now the classification of enzyme, there is several kind of enzyme included: oxidoreductase, hydrolase, isomerase, transferase, lyase. There are this function of enzymes are different groups of enzymes. Within these groups, different kinds of enzymes. In these groups, several enzymes are there. In this several enzymes are there. Several functions which some enzymes add the water groups in a particular product, and when some ligase we also broke the fatty acid structure. So several enzyme function is different group. There is different types of enzymes. So this is the ultimate classification of enzyme: oxidoreductase, hydrolase, isomerase, transferase, lyase, and ligase. And now how the structure of enzyme. What is the made of enzyme? Enzyme is basically made of with protein structure. You already know. And there is also non-protein structure. Non-protein structure, metals, ions, etc. are made of it. The non-protein part of the enzyme is called the cofactor. This cofactor is the non-protein factor. There is a protein part of the enzyme that is called the apoenzyme. Or another is the without the protein part. There is the part that is non-protein is